within 15 or 20 minutes lose all its activity. So how can you overcome this problem? How can you find a better biological part? Well, one strategy is that you simply go ahead and you screen for biological diversity. So in other words, you try to express and test homologs of this enzyme. And so lo and behold, by using this strategy, we identified a homolog in a marine archaeon called Nitrosopomonas maritimus, which shared 50 or 60% of sequence identity, but was approximately 100 times more stable under oxy conditions. So looking for biological parts and screening different enzyme homologs can help you to identify enzymes. But sometimes you have the problem that you would like to employ a certain enzyme reaction, but you actually don't have an enzyme in the database. And this is shown here, where you want to use a reaction in which a compound called methylsuccinyl-CoA is oxidized to become a compound called mesacenyl-CoA. Well, there had been enzymes described that can do this chemistry. The problem is that these enzymes are so-called dehydrogenases. And these dehydrogenases are very complicated enzymes because they oxidize the substrate, put the electrons onto a cofactor, and from this cofactor the electrons are put on a second protein called electron transfer flavoprotein, and from there on the electrons are put into a membrane-bound respiratory chain. So in other words, if you want to use this kind of chemistry, you need not only the enzyme, but you also need its interaction partner, and you need a membrane-bound respiratory chain, which is awfully complicated. So in this case, it might be helpful if you simply shortcut this whole electron transfer relay. And what we intended to do is to convert this methylsuccinyl-CoA dehydrogenase into a methylsuccinyl-CoA oxidase, an enzyme that could directly use oxygen instead of having this complicated electron transfer chemistry. So what you need to do here is you need to look into the active site of the enzyme, and you need to think about ways how to, for instance, accommodate an oxygen molecule there. So you can open up the active site by creating a bit more space. You can think about adopting and kind of allowing for a negatively charged to be developed, so putting more positively charged amino acids in the active site. And then you simply can go ahead and can test different active site mutations. And when we did this, we actually ended up and identified a triple mutant that could indeed catalyze the direct oxidation of methylsuccinyl-CoA in the presence of molecular oxygen. Or in other words, what we did here is we basically created our own enzyme for our own pathway. The question now is, at the end of the day, we had to identify 15 different enzymes from very different organisms. So we had enzymes in there from Methylobacterium extorquens. It's a plant organism, plant-associated organism. We had enzymes that we would take from Rhodobacter spheroides, an organism we would find in ponds. We would have enzymes that we've taken from Clostridium, which is a human pathogen. We have enzymes that were from E. coli, a well-known lab organism. We would actually have enzymes from Nitrosopomonas maritimus, a marine archaeon, and even an enzyme from the human liver, and on top of it, our re-engineered enzyme that catalyzes the oxidation of methylsuccinyl-CoA. The big question is now, can you bring all these enzymes together, and would they be able to fix carbon dioxide in an efficient manner? So we've done the experiment. So we've used an Eppendorf cup, and we've simply pipetted all 15 enzymes into this Eppendorf. We've added ATP and NADPH, and we've also added this green compound down here, which is called propionate-CoA. The idea being, if the cycle is active, it would start to turn, and for each time it starts to turn, it would actually accumulate a carbonate, which would be the direct carboxylation product. So in other words, if you follow that concentration over time, you would be able to see if the cycle is active or not. And as you can see here, indeed, we could demonstrate that the catch cycle fixes carbon dioxide over time, and actually at a rate of 0.2 CO2 molecules per acceptor. And it really demonstrates to you that you can come up with a theoretical pathway on the paper. You can look for the different enzymes, or even engineer your own enzymes. You can bring these enzymes together, and such a cycle would be functional. So you can design a biological systems just by using chemical criteria. Yet I hope you also realize that the cycle is not turning perfectly. So the rate is actually per acceptor an hour, and we all know that biological systems are operating at smaller time scales. So they should be operating per minute or per second. So the question here now is, why is the cycle not turning faster? So in other words, which design principle did we miss? This brings me to the second part. I'll tell you how we can optimize such metabolic effects. biological design principles. Or in other words, it's not enough to think about the biochemistry and the chemistry of enzymes and metabolic networks, but it's also important to think about the biology. So rephrasing the question, why is the cycle not turning faster? What actually limits CO2 fixation efficiency of the cycle? And the answer is very simple, and the answer is not very surprising. The point is that we've brought together very different enzymes from very different biological backgrounds. And these enzymes do not have a common evolutionary history, and these enzymes have not co-evolved together in this network, and these enzymes are doing things they're not supposed to do. In other words, these enzymes decide reactions, and the whole metabolic network accumulates dead-end products like malonyl-CoA, or for instance like malonyl-CoA. So to optimize now CO2 fixation efficiency, what we have to do is we have to go back and we have to debug the system. So we have to teach the system to be more specific. We have to teach the enzymes to work together. And we can apply very different principles. So one principle would, for instance, be that you can add more complexity to the system. So remember I talked about this compound called malonyl-CoA, which is a dead-end metabolite and which holds the cycle back from turning more efficiently. So there might be a way to actually remove this dead-end compound by adding another enzyme, which is so-called thioesterase. And this thioesterase would cleave malonyl-CoA into malate, as shown here, and it would release the CoA residue, which could be fed back into the cycle. So in other words, by increasing complexity of the cycle, by adding an additional enzyme that would proofread and scavenge and recycle the standard metabolite, we would be able to improve CO2 fixation efficiency. And in fact, what you can see, as soon as you add this thioesterase, you ramp up CO2 fixation efficiency. I think this is a very important principle, this metabolic proofreading, that we can use in cellular biology. But I think it's also important to realize that in many biological systems, such metabolic proofreading principles are also operating. So that's one way, metabolic proofreading, to improve efficiency. Another way we can use is basically redesign the original strategy. And we've used this, for instance, to circumvent a reaction from 4-kilo-CoA to this compound here called methylamine-CoA. This was not the reaction that we could use because it made too many problems. So we had to go back and detour the whole reaction by going to a compound called acryl-CoA, and from there on then back to this compound methylamine-CoA. The pathway redesign was very useful to us to identify enzymes that could do the job. And one other part of this design principle is, of course, to engineer enzymes to be more specific. And we've used this, for instance, to improve and get rid of a side reaction up here. So in other words, pathway redesign and enzyme engineering can help us also to improve CO2 fixation efficiently again. And finally, playing around with the physical conditions, for instance, improving and optimizing buffer concentration, and playing around with ionic strength can also help us then to finally speed up CO2 fixation efficiency. So at the end of the day, without changing the chemistry of this cycle, just by simply optimizing the enzymes, by adding metabolic proofreading, enzyme engineering, and slight pathway redesign, we end up with a cycle which was now turning at a rate of 0.6 CO2 molecules per acceptor hour. In other words, implementing biological design principles has helped us to increase CO2 fixation efficiency of the whole system by almost a factor of 20. I guess what I wanted to say here is that you can really think about having really good individual players. You can really think about getting the best enzymes together. And it's a little bit like the Dutch national team. There's really good individual players. But at the end of the day, you need to form a team if you want to win championships. In other words, it's always important to think about the chemistry, the biochemistry of the enzymes, but it's also important to think about the enzymes working together efficiently. So this is where we're standing right now with the catch cycle in version 5.4. The catch cycle now features 17 different enzymes from 9 different organisms, including 3 really generic enzymes. And it fixes carbon dioxide of a rate of 5 nanomoles per minute per milligram of protein. This is actually something that you would be able to measure if you, for instance, try to measure other CO2-fixing, natural CO2-fixing organisms in vitro. So with this system, we are almost very close to biological reality, at least in vitro. Of course, the catch cycle is not this version 5.4, the last version, but I think it's not the end. I think it's a starting point for several research directions. And one thing we have to do is entering the so-called design, build, test, and analyze cycle, which is a classical improvement that you can use in synthetic biology. So the idea would be here now to test different principles. So where are the boundary conditions of the system? How much can it be improved when you optimize enzyme concentrations and metabolite concentrations? How can it improve when you, for instance, add allosteric regulation? Can it be improved if we try to implement metabolite channeling? And would it actually be improved if you, for instance, try to implement something like compartmentalization? And I think through iterative rounds of reconstructing the system by changing parameters, would be able to improve the catch cycle even further. What are the next steps once we have improved catch cycle? So one idea would be to use the cycle in a true bottom-up fashion. So using it as a metabolic module that we, for instance, could use to implement into cell-like structures, artificial cells, membranes, liposomes, where we can, for instance, feed in light, we can feed in hydrogen or electricity, where we can energize the system and use our metabolic module to fix cause if you want to say so, for instance, developing an artificial organelle, an artificial clone. And of course, another thing we can do is we can try to implement and try to transplant the catch cycle in a top-down fashion into living systems. So the in vivo transplantation is one of the big challenges that lies ahead of us. I hope we can also understand that a complex network like the catch cycle that consists of 17 different enzymes might not be very easily transplanted into a living system that features 3,000 different reactions. And we expect that this will be a challenge of synthetic biology. Okay, so this is the end of my lecture. I hope I could show you that synthetic metabolism is really something that we can achieve in future in synthetic biology. I can show you that synthetic metabolism is built on the concept of being freely able to choose and design theoretical pathways on the paper. I hope I could show you that synthetic biology involves finding engineered enzymes to realize this cycle in the lab, but it also involves building an optimization of the pathway sequence and iterative rounds. I could show you that one of the future steps in synthetic metabolism will be to transplant such artificial cycles into living cells, let it be natural or artificial cells. That's the end of my talk, and I would not be standing here without a great number of great scientists in my lab. I just want to highlight Thomas Schwander, who actually built the catch cycle, the first proof of principle. I want to highlight those who have been optimizing the biochemistry of anaerobic carboxylase reactants. But I should not forget the funding agencies, and I thank you for your attention.
Hi, my name is David Picard, and I'm the head of the Synthetic Biology Group at the Pasteur Institute in Paris. And today I'm going to talk to you about CRISPR technologies for bacteria and some of the work we're doing in my lab. But first I want to give you a brief history of CRISPR system and how these fascinating systems were discovered. And the story begins in 1987 when a Japanese group made this interesting observation where they sequenced a piece of the Escherichia coli chromosome, and next to this IAP gene, they identified this interesting locus with repeated sequences that were interspaced by these variable sequences here, but that had a very conserved size. And it's only much later that we really understood what these systems were actually doing. And in basically the next 25 years, with the advent of genome sequencing, we started to have a lot more sequences of archaeal and bacterial genomes, and people were identifying these interesting loci in a lot of places. And they were identified, developed many times and given many different names. DVR, TREP, LTRR, and the list goes on. And it's only in 2002 that basically the name CRISPR was coined and unified the field, and this stands for Clustered Regularly Interspaced Short Palindromic Repeats. And in this paper by Jansen in 2002, they also described the existence of a set of genes that were constantly associated with this CRISPR loci, and this name and Cas genes for CRISPR associated here. So the way this loci look is you have the CRISPR locus itself with the repeats and spacers, and the Cas genes that are very frequently found next to this CRISPR. It's only in 2005 that 3 papers were published the same year making the same discovery that the spacer sequences between the repeats, some of them are actually matching foreign DNA and specifically bacteriophage DNA. So you have these 3 papers that made this observation, and in particular also the paper from Francesco Morica, where they found this to be consistently the fact in many bacterial and archaeal species, and they also observed that bacteria that carried CRISPR spacers that were matching a bacteriophage could not usually be infected by that bacteriophage. But this was still all theoretical and bioinformatics work, and the experimental evidence that CRISPR system was actually an immune system to defend against foreign elements and bacteriophages only came with this science paper in 2007 by Rodolphe Barambou in the group of Philippe Horvat. And this is an interesting paper in many respects, and in particular because it comes not from academia, but from an industry and a company called Danisco, where they produce ferments for cheese and yogurt production. And this industry, they always need to make sure that the strain, bacterial strain they're using cannot be infected by bacteriophages. They make milk cultures of very large volume, thousands of liters, and it can cost a lot if you get bacteriophage contamination. And so when selecting strains, and specifically this paper, it was Streptococcus thermophilus that are resistant to bacteriophages, they found that the CRISPR locus was actually changing and adding novel repeat spacers units. And specifically, they did this experiment where they infected the strain with 2 different bacteriophages, and they noticed that the CRISPR system was capturing novel sequences that matched the genome of these bacteriophages, and then it would become resistant to the corresponding phage. So this was the first experimental evidence that CRISPR systems are an adaptive immune system of prokaryotes. In the next years, many groups started to look into these CRISPR systems and describe their mechanism and mode of action. And basically, when a bacteriophage injects its DNA into the cell, then a group of Cas protein, and specifically Cas1 and 2, are involved in capturing pieces of DNA from the bacteriophage and integrate it as a novel spacer in the CRISPR locus. The CRISPR locus is then transcribed, and the transcript is then processed into these small guide RNA or CRISPR RNA that are able to guide a complex of Cas proteins to go and target and destroy target sequences. So the first step is referred to as immunization or CRISPR adaptation, CRISPR spacer acquisition, and then we have the immunity phase of CRISPR. You need to know that CRISPR come in many shapes and sizes. They've been classified now in 2 classes, class 1 and class 2. Class 1 systems have an effector complex of Cas protein that consists in many different proteins that you see here, while Cas 2 systems have just one protein that is able to carry out the immunity interference step. But then class 1 and class 2 can then be further subdivided into many types and subtypes of CRISPR systems. And here I'm not going to go through all this in detail, but basically there are around 20 subtypes that have been described of all this CRISPR system. And very interestingly, they all differ in the way they carry out many aspects of the CRISPR biology, including the RNA processing and the interference. And it's also important to know that some of these CRISPR systems target DNA, but others are able to target both DNA and RNA, and some seem to only target RNA. But here, we're mostly going to talk about type 2 CRISPR system, and specifically type 2A CRISPR system, because those are the ones that are mostly used right now for all the biotechnological applications. And specifically, this system from Streptococcus pyogenes, which was described in the first place by the group of Emmanuel Charpentier, is an interesting one. So it carries a CRISPR array here, and this set of 4 Cas genes, and Cas9 is the only one required for the interference step. But the way it works is that the CRISPR is first transcribed into this precursor RNA, but another RNA is also produced by the system. And this RNA here, the tracer RNA for transacting CRISPR RNA, carries a sequence that's homologous to the repeats in the CRISPR array. So it's able to form this duplex RNA that is recognized by Cas9 and by the host RNA tree, and that will process it to form the final guide RNA, also called the CRISPR RNA. So you form this nucleoprotein complex that basically acts as a surveillance complex in the cell. It's constantly scanning the DNA present in the cell, and the way it does that is by first looking for a small sequence motif known as a protospacer adjacent motif. In the case of this specific Cas9, the motif is just a GG dinucleotide. When it finds this GG dinucleotide, it then starts to unwind the DNA and try to pair it with a CRISPR RNA. If that can be done successfully, it will trigger a conformational shift in the Cas9 protein, bringing 2 catalytic domains in contact with the DNA and introducing a double-strand break. And this leads to the target destruction. So all this system has been described mostly in the papers listed here that you can have a look at. And this discovery led to a lot of interest, because being able to target a nuclease to a specific position by just changing the sequence of RNA here is something that's very interesting for many biotechnological applications. And the year basically after the system was really described in vitro, so in 2013 was what people call the CRISPR craze, and many papers came out describing the use of this Cas9 nuclease to modify genomes. And so I was happy to contribute to the work that was done to demonstrate this in bacterial genomes, Streptococcus pneumoniae, and E. coli. But you see all this list of papers where they did that in human cells, mice, frogs, zebrafish, and the list goes on. So now you basically have CRISPR tools for all your favorite organisms. The way these technologies work always basically starts by reprogramming the Cas9 nuclease, but with a guide RNA to target a position of interest, and this will introduce a break in the DNA. And then you can have different possible outcomes. If you provide a template DNA that can be used to repair the break through homologous recombination, you can use this to introduce many types of mutations, point mutation, insertions, deletions. You can very accurately modify the target sequence. If you don't provide a template for homologous recombination, but if your cells are able to carry another type of repair pathway that's called non-homologous enjoining, or NHEJ, then this NHEJ pathway can repair the break, but does that in a very error-prone way, very frequently making mistakes, insertion and deletions, indels, and that can be very useful also to introduce frame shifts in target genes and knockout genes. Also, there is a third possible pathway, which is if the cell is not able to repair the break, then